I was just reading the latest clinical data on purinergic sleep signaling, and it changes how we should evaluate homeostatic sleep pressure in healthy aging adult populations. Many people notice a heavy, unavoidable urge to sleep after a long day, assuming it is controlled entirely by the time on the clock as we navigate healthy aging during daily cognitive tasks. We commonly treat sleepiness as a simple scheduling response, without considering the specific chemical molecules that build up in our brain tissue in our daily lives to protect our brain structure. However, neuroscience suggests that this sleep pressure is driven by the progressive accumulation of a neurochemical called adenosine during waking hours within neural tissues as we navigate healthy aging. Imagine a biological metabolic meter that tracks every minute you are awake. By slowly piling up signaling molecules in the synaptic space under these metabolic conditions in our daily lives. We are exploring how adenosine signaling coordinates sleep drive, helping to reframe sleep pressure from a scheduling habit to a cellular clearance mechanism to support cognitive endurance within neural tissues. When adenosine levels rise without sufficient clearance, cognitive speed declines, reaction times slow, and maintaining focus becomes increasingly difficult as we age over time under these metabolic conditions. If we try to override this sleepiness using caffeine, we block the receptors temporarily, but do not clear the accumulating chemical signal in healthy adult populations to support cognitive endurance. The stakes are high because chronic accumulation of adenosine without deep sleep clearance is associated with reduced synaptic plasticity and cognitive fatigue for long-term health span as we age over time. Understanding the physical pathways of adenosine signaling allows us to design practical, low-risk protocols that support our sleep-wake cycles naturally during daily cognitive tasks in healthy adult populations. The problem is that modern lifestyles often disrupt the natural balance between adenosine accumulation and the deep sleep stages required for clearance to protect our brain structure for long-term health span. By identifying the specific molecular mechanisms of purinergic signaling, we can optimize our daily routines to support long-term cognitive endurance as we navigate healthy aging during daily cognitive tasks. To understand this neurochemical process, we must define adenosine, a purine nucleoside that acts as a central nervous system depressant and regulator in our daily lives to protect our brain structure. Adenosine is produced as a natural byproduct of cellular energy consumption, specifically from the breakdown of adenosine triphosphate, or ATP, within neural tissues as we navigate healthy aging. During waking hours, active brain cells consume ATP constantly to support synaptic transmission, releasing adenosine into the local extracellular space under these metabolic conditions in our daily lives. This extracellular adenosine binds to specific purinergic receptors, primarily the A1 and A2A receptors, expressed on neurons throughout the brain to support cognitive endurance within neural tissues. Binding to these receptors inhibits neural activity, gradually reducing the overall excitability of brain networks as the day progresses over time as we age over time under these metabolic conditions. This progressive inhibition creates homeostatic sleep pressure, which is the biological drive to sleep that increases with every hour of wakefulness in healthy adult populations to support cognitive endurance. This homeostatic sleep drive operates independently of the circadian rhythm, which is the internal clock that regulates the timing of sleep for long-term health span as we age over time. During healthy sleep, the brain actively clears extracellular adenosine, restoring receptors to an unbound state before the next waking cycle begins during daily cognitive tasks in healthy adult populations. As we remain awake, the continuous breakdown of ATP leads to a steady rise in adenosine concentration in the basal forebrain to protect our brain structure for long-term health span. This accumulation activates A1 receptors, which hyperpolarize neurons and reduce the release of excitatory neurotransmitters like glutamate and acetylcholine as we navigate healthy aging during daily cognitive tasks. This reduction in excitatory signaling dampens the activity of arousal centers, making it increasingly difficult to sustain high levels of cognitive focus in our daily lives. Simultaneously, adenosine binds to A2A receptors in the striatum, activating sleep-promoting networks that coordinate the transition from wakefulness to sleep within neural tissues. This dual action on arousal and sleep networks ensures that the drive to sleep matches the brain's metabolic expenditure during wakefulness under these metabolic conditions. Caffeine functions as a structural analog to adenosine, binding to these same purinergic receptors without activating the inhibitory signaling pathways to support cognitive endurance. By blocking the receptors, caffeine temporarily prevents the brain from detecting the accumulating adenosine, suppressing the felt experience of sleep pressure as we age over time. However, adenosine production continues, creating a silent backlog of signaling molecules that remains in the extracellular space during caffeine use in healthy adult populations. Once the caffeine is metabolized, this backlog binds to the newly vacant receptors, causing a sudden and severe drop in cognitive energy for long-term health span. This sudden drop, known as a caffeine crash, illustrates that masking the signal does not resolve the brain's physical need for clearance during daily cognitive tasks. During deep, slow-wave sleep, the brain's glymphatic system increases the flow of interstitial fluid to wash away accumulated adenosine and metabolic waste to protect our brain structure. This nocturnal clearance process resets the homeostatic sleep meter, ensuring we wake up feeling alert and mentally clear the next morning as we navigate healthy aging. If slow-wave sleep is disrupted or shortened, adenosine clearance is incomplete, leaving a residual chemical load on the receptors at wakefulness in our daily lives. This residual load causes immediate morning grogginess and reduces cognitive stamina, forcing the brain to operate under constant neurochemical inhibition within neural tissues. The physical maintenance of synaptic pathways is closely dependent on the complete clearance of these purinergic signaling molecules during sleep cycles under these metabolic conditions. One limitation of adenosine research is the difficulty of measuring real-time extracellular chemical concentrations in the brains of living human subjects to support cognitive endurance. We rely on indirect measures such as sleep EEG patterns, caffeine response studies, and positron emission tomography imaging of receptor binding as we age over time. In clinical cohorts, individual sensitivity to caffeine varies significantly due to genetic variations in receptor structure and liver metabolic enzymes in healthy adult populations. We must also recognize that sleepiness is not regulated solely by adenosine, as circadian signals and ambient temperature also influence drive for long-term health span. The population boundaries of current studies are narrow, and much of our data relies on controlled laboratory sleep deprivation protocols during daily cognitive tasks. Because we cannot directly observe real-time glymphatic clearance in humans, we must interpret sleep quality and hormone assays with scientific caution to protect our brain structure. Therefore, we analyze adenosine accumulation as a key contributing mechanism in sleep regulation rather than a single indicator of sleep quality as we navigate healthy aging. Answering our opening question, the urge to sleep is driven by a specific chemical buildup rather than a simple scheduling choice in our daily lives. By understanding this physical mechanism, we can manage our daily energy better, focusing on proper clearance rather than masking the signal within neural tissues. When slow-wave sleep is protected, adenosine clearance is complete, which helps maintain synaptic efficiency and cognitive focus during waking hours under these metabolic conditions. The practical challenge is supporting natural sleep architecture through low-risk behavioral habits and consistent routines as we navigate healthy aging to support cognitive endurance. While we cannot modify the rate of adenosine production, we can optimize the environmental factors that support deep, slow-wave sleep clearance as we age over time. Limiting caffeine consumption to the early hours of the day prevents receptor blockage from disrupting the natural onset of deep sleep in healthy adult populations. Maintaining a consistent sleep schedule helps align homeostatic sleep pressure with circadian timing signals, promoting efficient sleep architecture and clearance for long-term health span. These behavioral habits do not change neural connections directly, but they support the chemical cleaning process required for optimal synaptic performance during daily cognitive tasks. Supporting adenosine clearance requires a unified focus on sleep consistency, early caffeine limits, and proper wind-down routines to optimize these pathways to protect our brain structure. To apply these concepts, limit caffeine intake to the morning hours, ensuring at least 10 hours of caffeine-free time before sleep as we navigate healthy aging. Establish a consistent sleep schedule by going to bed and waking up at the same time daily to support natural sleep architecture in our daily lives. Prioritize a quiet, cool, and dark bedroom environment to facilitate the deep, slow-wave sleep stages necessary for efficient lymphatic clearance within neural tissues. Consult a healthcare professional if you experience chronic sleep disruption, as persistent sleep issues can impact cognitive function and overall health under these metabolic conditions. Avoid relying on high-dose sleep aids or stimulants to manage your sleep-wake cycle, as they can disrupt natural sleep stages and clearance to support cognitive endurance. Please remember that this analysis is intended for educational purposes and should not replace individualized clinical evaluation or specific medical advice as we age over time. Focus on establishing consistent, low-risk daily habits that support natural sleep drive and reduce systemic metabolic stress naturally and safely in healthy adult populations. Track your sleep consistency, daily caffeine timing, and morning alertness to build a personalized, evidence-bounded framework that supports your long-term cognitive health span for long-term health span. Optimize the protocol, protect your brain, and I'll see you in the next data set.